So the next one is in the phospholipids, that is lecithin. So how this lecithin is made up of? Okay, this lecithin is made up of alcohol plus fatty acid, phosphatidic acid. So as I said, phosphatidic acid base is for synthesis of rest of the glycerophospholipids. Okay, and it is having a group choline. The nitrogen base present here is a choline. Okay, the nitrogen base present here is a choline. So where and all it present? This phosphatidyl lecithin is present in brain, nervous tissue, sperms and egg yolk. Okay, sperms and egg yolk. The main importance of lecithin is they are surface active agent and helps in emulsification of fats. So when you are consuming a diet which is rich of fats, okay, it should also have lecithin. So they are very much helpful in emulsification of fats. So surface active agents. And the important point is dipalmityl lecithin, okay, which is a lung surfactant. So what is the use of lung surfactant? Which is lowering your lung surface tension and expands the lung volume. Okay, so that means by expanding the lung volume, it prevents the collapsing of the lung alveoli, because whenever you are inhaling the air, your lungs has to be bulged. Okay, and when you are expiring, your lungs has to be collapsed, right? Again, for the expansion, okay, so they shouldn't be flattened, they shouldn't stick together. So for that, this dipalmityl lecithin acting as a surfactant agent, okay, which will not allow the lungs to flat, okay, get collapse, they are not allowed to be flattened. Okay, so for this expansion and collapsing, okay, there is a medium barrier that is called dipalmityl lecithin. So because of lack of this dipalmityl lecithin, there is a serious disorder, respiratory distress syndrome in children, especially in infants. Okay, we'll discuss separate topic as, okay. So now, absence of dipalmityl lecithin, that's what I was mentioning, okay, in premature infants, it may produce respiratory distress syndrome or hyaline membrane disease. Okay, you see here the structure of lecithin when you come across. Okay, I will highlight. So this part is glycerol, yellow. Okay, and R1, this part is fatty acid. Okay, and this part is phosphate group with the nitrogen base. Okay, that is choline. Okay, this part is choline group, CH2, CH2N and 3 methyl groups. Okay, that is nitrogen base choline. So otherwise this lecithin otherwise called as phosphatidylcholine, as it is having additional group choline. So this lecithin otherwise called as phosphatidylcholine. So we have already come across the importance of this lecithin or choline in the diet. Okay, next coming to cephalins. Cephalin, so the composition of cephalin is alcohol. Okay, because the standard alcohol, because we are talking about glycerophospholipids, so the standard alcohol is glycerol and fatty acid plus phosphoric acid. Okay, and ethanolamine or serine. Okay, the substance is ethanolamine or serine. Okay, or serine means there is a separate amino acid is there. Okay, so the nitrogen base is provided by ethanolamine or serine. Okay, where and all it presents? It present in brain, erythrocytes and many other tissues. You see here, so this part is a nitrogen base which is called as phosphatidyl okay, or else is called as cephalin. So phosphatidylinositol, okay, so phosphatidylinositol, again its composition phosphatidic acid, that means phosphate group is there, 2 fatty acids are there and glycerol is there, and nitrogen as a nitrogen base. Okay, in place of nitrogen base, okay, we have inositol. Okay, alcohol, that's type of carbohydrate. Inositol is a type of carbohydrate, alcoholic form of carbohydrate in place of nitrogen base. Okay, they are very, very important for cell membrane formation. What is the importance of this phosphatidylinositol? They are important components of cell membrane. Okay, without this there is no formation of cell membrane. Okay, and certain hormones such as oxytocin, vasopressin, so they're all mediated through phosphatidylinositol. So that means phosphatidylinositol acting as a secondary messenger for several hormones. What are the hormones? Oxytocin and vasopressin. Okay, oxytocin and vasopressin. Oxytocin, where do you require? Okay, this is a hormone during the labor. Okay, contraction of uterine muscles. Okay, you do require oxytocin. Okay, and vasopressin is antidiuretic hormone. Okay, so all these two for their action they require phosphatidylinositol. Without phosphatidylinositol there is no action of these hormones. Okay, in response to hormonal action, PI is cleaved to diacylglycerol and inositol triphosphate. Okay, this phosphatidylinositol cleaved to diacylglycerol and inositol triphosphate. So this is the structure. Here you see this is a carbohydrate form. Okay, what we are mentioning? Alcoholic form of carbohydrate. Okay, along with the phosphate group. Okay, along with the phosphate group. So now phosphatidylserine. If amino acid, okay, serine is present with the glycerophospholipids, that phospholipid is known as phosphatidylserine. So amino acid serine you see here. Here the source of nitrogen base coming from amino acid serine. So like this, NH3CH, COOH and R. So serine that is like CH2OH. So this is phosphatidylserine. This CH2OH involved in phosphodiester linkage with the phosphate group in the phosphatidylserine. So plasmalogen. So plasmalogen, what do you think of plasmalogen? Plasmalogen, they differ from lecithin or cephalin at first position of glycerol, where fatty acid replaced by long chain unsaturated aliphatic aldehyde such as palmitic acid or stearic aldehyde. Okay, so far at the first carbon of glycerol we used to get attached by unsaturated fatty acids or saturated fatty acid, but here the long chain unsaturated aldehydes, okay, long chain unsaturated aliphatic aldehydes used to be attached. And plasmalogen presence, where and all present? These plasmalogens present in skeletal muscles, okay, and as well as in cardiac muscles and in semen. The location of plasmalogens, they are where and all they present? They are all present in semen, skeletal muscles, okay, and in cardiac muscles. You see here at the first carbon of glycerol, long chain unsaturated aldehydes, okay, aliphatic aldehydes, okay, long chain unsaturated aliphatic aldehyde attached in place of, here you can see in place of ester linkage you are seeing here ether linkage, okay, here you can see ether linkage. That is the difference of plasmalogen among all glycerophospholipids. Cardiolipins, this is simply like it is a diphosphatidylglycerol. That means here not a single glycerol molecule, but it is attached with the double glycerol molecule. That means two glycerol molecules are attached, okay. It contains two molecules of phosphatidic acid held by glycerol, okay. It is attached it is attached by two molecules of phosphatidic acid. That means two molecules of phosphatidic acid, you know, already two glycerols are there in phosphatidic acid, I mean two molecules, and these two phosphatidic acid attached by a single glycerol molecule. So how many glycerols are there? Totally they are having three glycerol molecules in the structure, but only pure glycerol is there in the middle, okay, and rest of the two glycerols involved in making of phosphatidic acid, okay. One glycerol is a pure form of glycerol which holds the other two phosphatidic acids, okay. And where and all it present? It present in inner mitochondrial membrane and has antigenic properties, okay. Cardiolipin has got strong antigenic properties. Ceramide. So what is a ceramide? So the esterification of sphingosine with a fatty acid or high molecular weight. That means when high molecular weight alcohol in place of glycerol, the alcohol sphingosine esterified with a fatty acid, okay, which is a high molecular weight one, okay. Alcohol sphingosine attached with a high molecular weight fatty acid by ester linkage, and they are all found in brains and nerves. These ceramides, okay, all get in the brain and nerves. And which is common here? The ceramide is common for all glycolipids and sphingomyelins. Okay, so as we mentioned, the location brain and nerves. So the glycolipid which is present in the cell membrane, okay, or in the tissues, okay, or in the nerves, brain cells, where they required is ceramide. Sphingomyelin. So as I have discussed in the beginning, okay, with the name itself, sphingophospholipids, otherwise known as sphingomyelins. Why? Because the location of sphingophospholipids mainly in myelin sheath of neurons, okay, neuronal cells. So based on this, sphingophospholipids, otherwise known as sphingomyelins, okay, sphingomyelins. So it doesn't contain glycerol, but it contains unsaturated amino acid alcohol, that is sphingosine. Okay, it contains what? It's a contain unsaturated amino alcohol, that is sphingosine. And what are the things it contains? It may contain choline. Okay, it may contain choline, phosphoric acid and fatty acid. Okay, it contains choline, phosphoric acid and a fatty acid. Sphingomyelins can make large part of myelin sheath, as I mentioned in the beginning of sphingophospholipids, right? So they play major part in formation of myelin sheath. Okay, and the location: brain, lungs, nerve and other tissues. The location of sphingomyelins, okay, where and all they located? Brain, lungs, nerves and other tissues. And the deposition of sphingomyelin in liver and various parts of the body like lymph nodes, bone marrow and central nervous system results in a lipid storage disorder, Niemann-Pick disease. Note down this point. Niemann-Pick disease is a type of disease, comes under lipid storage disorder. It is a inborn error of lipid storage disorders. Okay, inborn error of lipid metabolism, Niemann-Pick disease. Why? Because so as sphingomyelins are located in myelin sheath, okay, during the normal turn and wear of the lifespan, okay, after completing of lifespan, they should be degraded. These these sphingomyelins are degraded by the enzyme sphingomyelinase in these tissues. Okay, if genetically there is error in the enzyme sphingomyelinase, so whatever the sphingomyelins in the concerned tissues, they cannot be degraded. So what happens if they are not degraded? They start accumulating in particular parts and they cause a lipid storage disorder, Niemann-Pick disease. It is one of the rarest disease and comes under genetically inborn error, okay, because of the absence of the enzyme sphingomyelinase. That is the significance of sphingomyelins. So sphingophospholipids, you see the structure in this slide. Okay, sphingomyelins made up of a ceramide, okay, and a phosphorylcholine and a fatty acid. Three parts. Okay, three parts it is making of. First part is ceramide. Okay, second part is phosphorylcholine and third part is fatty acid. Okay, three parts is made up of ceramide, phosphorylcholine and fatty acid. So what happens? Sphingomyelinase enzyme, it breaks down, hydrolyzes or break down the sphingomyelin into phosphorylcholine and ceramide and fatty acid. Okay, so if this is not there, sphingomyelin is going to accumulate in the tissues, liver cells or in lymph nodes and causing a disease, Niemann-Pick disease.